Hi, I'm Dimitrios Evangelopoulos, and my project title is Investigation of Drug Synergy and Rational Design of Drug Combinations. Antibiotic resistance is one of the biggest public health challenges of our time. Infections that once could be treated can now be fatal. With a limited number of new antibacterial drugs being developed, it is important to identify ways to maximize the lifetime of the existing drugs, to save our drugs. In our research project, we study the use of different drug combinations to kill resistant bacteria. Combination therapy will become the norm in the near future, as treating bacterial infections with a single antibiotic will be less and less frequent due to the current resistance making them redundant. We are planning to identify drugs that work better in a combination than in isolation. To achieve this, we will first test the capacity of single drugs and drug combinations to kill bacteria in the lab. We will then examine the development of resistance to drug combinations using the best identified combinations in a model of human infection known as the hollow fiber system, seen here in the bottom left. A simple cartridge containing several tiny tubes where bacteria and drug amounts can mimic what really happens inside our bodies. But we also want to find out what the drug combination treatment means for the bacteria. And for this, we're going to monitor the changes in the dynamics of gene expression from DNA to RNA, and also analyzing the chemical processes by measuring the wide number of metabolic products in the bacterial cells known as metabolites. And this is what you can see in the panels in the bottom right, where single drugs and drug combinations have a completely different response, and the different metabolic pathways that are involved are different. This will look like trying to find out the bacterial traffic map system and the way they interact during a drug combination therapy. Think of trying to identify which traffic lights go on and which are going off based on the different drug treatments. Throughout this project, we will be able to predict which drug combinations will work best and how these combinations achieve superior bacterial killing. But most importantly, our findings will contribute towards the increased clinical lifetimes of our current and viable antibiotics. Thank you.